Hello everyone, my name is Swati Shallika and today we will discuss about salting up. So when designing a purification scheme for protein or enzyme, generally centrifugation is the first step. Okay, and salting out is second step following centrifugation. So I have already told you about different methods for purification of protein in my previous video, and here we will discuss only about salting out. So salting out is basically a separation technique where separation takes place on the basis of solubility of the protein. Okay, so in this video we'll discuss 3 points. First is what is a protein molecule and what is salting out, and third is what property of protein it uses. Okay, so let's begin with what is protein. So we all know that proteins are made up of amino acids, and there are number of amino acids which is present, and some of them are hydrophilic in nature and some of them are hydrophobic in nature. So what will be the arrangement of this hydrophilic amino acid and hydrophobic amino acid when we put a protein molecule into a water solution? Okay, let me draw a diagram. Let me zoom in the camera. Okay, suppose this is the protein molecule. What will be the arrangement of hydrophilic amino acid? It will present on the surface of the protein molecule. Here, this is hydrophilic amino acid, and all the hydrophobic amino acid will be present inside the protein molecule. Proteins are soluble in any kind of aqueous solution or water solution because of these hydrophilic amino acid side chain. Okay, they interact with the water molecule, they interact with the polar molecule, and in this way they get solubilized into the water. Now I will tell you about 2 different cases. Okay, so that you will understand better what is salting out actually is. So first, let me draw a diagram. This is case number 1 without the salt. Okay, suppose this is a beaker which is filled with the water and you are adding protein into this. This is protein. What will happen there? What will happen here? It will acquire the same arrangement I told you here. Okay, all the hydrophilic molecules, all the hydrophilic amino acids will interact with the water molecule and form a hydrogen bond there. Okay, in this way proteins are soluble in aqueous solution. Now the second case, with the salt. Suppose this is a beaker which is filled with the water and we are adding protein plus salt. Generally we take ammonium sulfate. Generally we take ammonium sulfate. ठीक है, तो what will happen? As soon as we begin to add salt into this aqueous solution, suppose this is the salt molecule, black one is the salt molecule, okay. Black one is the salt molecule and blue one is the protein molecule. Okay. As soon as we begin to add the salt, what will begin to happen is the solubility of the protein in the water will begin to decrease. Okay. Not only that, if we continue adding more salt, eventually we are going to reach a certain specific concentration of salt, and at that value what happens is that protein will become insoluble in water. That means the protein is beginning to precipitate out of the solution and it will crystallize and form the solid state, and this is what we call salting out. Okay, protein will begin to precipitate like this. Okay, see the difference here. See the difference between these two cases. Clear? So when we add salt, the charged ions interact with water. This means that there are less soluble water molecules to interact with the protein. This one. Okay. As a result, protein molecules begin to aggregate like this, which causes precipitation. Okay. So this is all about salting out. Okay. तो बेसिकली सॉल्टिंग आउट में हम लोग क्या करते हैं, प्रोटीन को एग्रीगेट करते हैं, समहाउ प्रेसिपिटेट कराने की कोशिश करते हैं, सो दैट हम लोग उस प्रोटीन मिक्सचर को सेपरेट कर सके फ्रॉम अदर कंपाउंड. हाँ, तो दिस इज जस्ट द स्टेप. ओके, आई होप I made myself clear. Next video I will tell you about next method, that is dialysis. Okay, thank you.